Good afternoon and welcome back. I am Solomon. Today I am with new chapter 4, which focuses on cellular metabolism and metabolic disorder. This chapter is somehow broader, so I divided it into three parts. So in part 1, cellular metabolism, enzymes and their role in metabolism, chemical nature and classification of enzymes, mechanisms of enzymes actions, as well as factors affecting enzymatic activity are going to be seen in this video. So as usual, don't forget like, share, comment and subscribe to my channel. Thank you very much. Well, we are doing nice. So this is chapter 4, which focuses on cellular metabolism and metabolic disorder. This cellular metabolism is all chemical reactions that occur in living organisms. Therefore, we'll see in detail, especially respiration and photosynthesis as a chemical reactions, and in detail we'll see about enzymes, because every chemical reaction that occur in living organisms is catalyzed by enzyme. Therefore, it is a must to discuss about enzymes. And finally, we'll see some metabolic disorders, their diagnosis and treatment. So this is cellular metabolism. Macromolecules are formed and they can be broken down. Substances are transported across cell membrane either by active or passive transport. Genetic informations can be transmitted from parent to offspring, especially during reproduction. This and other activities takes place in living organisms. Most of these cellular activities require energy, a series of chemical reactions in a metabolism in which a product of one reaction is a substrate or a reactant for the next reaction is called metabolic pathway. For instance, if this is a metabolic pathway, A is the initial substrate or initial reactant, which is converted into product B by enzyme, that is a reaction catalyzed by enzyme 1, and this B, product of the first reaction, can be a reactant or the substrate for the second reaction, and it can be catalyzed by enzyme 2 and produced product C. Similarly, product C can be a substrate or can be a reactant for reaction 3, and that can be converted into the product D. This reaction is catalyzed by enzyme 3. All these activities or these pathways in general is called metabolic pathway. The cellular metabolism is divided into anabolism and catabolism, or this anabolism can be divided into anabolism and catabolism. Anabolism means anabolic reaction, or it can be endergonic reaction. Catabolism means catabolic reaction, or it can be exergonic reaction. So in general, metabolism can be divided into these two categories. Catabolic reaction releases energy. In catabolism or catabolic reaction, energy is released, whereas larger molecules are broken down into smaller ones. For instance, respiration is a good example of catabolism. In respiration, as you know, glucose, C6, H12, O6, glucose can be hydrolyzed or broken down into carbon dioxide plus carbon dioxide plus H2O, water, can be broken down or hydrolyzed into carbon dioxide plus water, but here the energy in the form of ATP is produced. This is a general formula for respiration. ATP is energy in the form of ATP is produced. Here oxygen may be added. Of course, respiration has different or can be divided into different levels, so I will see in detail where oxygen is used in respiration, but in general, in respiration, glucose molecule is hydrolyzed or broken down into carbon dioxide, water, and energy in the form of ATP is produced. Of course, this glucose is obtained from polysaccharides, for example, starch. Starch is a large, or it is a polysaccharide. So this starch can be hydrolyzed into glucose molecule. Therefore, glucose is hydrolyzed or respired or broken down into these molecules. In anabolic reaction, they need energy. Instead of release energy, anabolic reactions need energy, and in anabolic reaction, larger molecules are built from smaller molecules. So it seems it is a reverse of catabolism, because, for example, photosynthesis is a good example of anabolic reaction. In photosynthesis, for example, carbon dioxide plus water, water H2O, and then the presence of energy. Of course, sunlight is also required, chlorophyll is also required, but larger molecules such as glucose, and gradually glucose will be glucose molecules are joined together to make larger molecules such as starch. Therefore, in anabolic reaction, smaller molecules are joined together, then larger molecules are formed. Of course, they require energy. By the way, we'll see in detail how photosynthesis is carried out, especially in green plants, in this chapter. The reactants and products of metabolic reactions altogether are known as metabolites. These metabolites include, for example, amino acids, because amino acids are reactants for protein. Monosaccharides. These monosaccharides can be reactant for formation of other carbohydrates, for example, disaccharide and polysaccharide. Nucleotides are monomolecules for nucleic acids such as DNA and RNA. Proteins, which are products of amino acids. Carbohydrates, lipids, coenzymes, cofactors, DNA and RNA, these all are metabolites. Metabolites can be hydrophilic, that are polar, and hydrophobic, which are non-polar in nature. Polar metabolites are soluble in aqueous solution and include most sugars, purines, pyrimidines, nucleosides, acyl carotenoids, organic acids, hydrophilic acids, amino acids, phosphorylated compounds. These all are polar metabolites, whereas non-polar metabolites or hydrophobic metabolites are commonly lipids. So they are functioning energy store, and they are main components of cell membrane structure, and they are used for signaling of, or they are used as a signal transaction. The next subtitle for this chapter is enzymes and their role in metabolism. Almost all chemical reactions within the cell need a catalyst to speed their rate. Catalysts are two types: biological catalysts as well as non-biological catalysts. Therefore, we only focus on biological catalysts. So these biological catalysts are enzymes, and some RNA types can be used as a catalyst. In some cases, enzymes can make chemical reactions million times faster than without them of the same reaction. Therefore, the function of enzymes in a chemical reaction is they speed up chemical reactions even million times faster. So enzymes speed up reaction by lowering activation energy. They speed up chemical reactions by lowering activation energy. This activation energy is an energy which is required to start or to begin a chemical reaction. For example, if we put the match and the stick together, we can't make any fire, but we need to apply some force, some friction force to start to make fire between the stick and the match. This the applied friction force or energy is it is an activation energy which is used to start or begin a chemical reaction. Therefore, the role of enzymes in a chemical reaction is to speed up a chemical reaction by lowering its activation energy. So the following are general properties of enzymes. For example, almost all enzymes are proteins, even if some enzymes need a non-protein part in their function, but almost all enzymes are proteins in nature. They are heat liable or they are heat sensitive molecules. Still, they are water soluble molecules. They are sensitive to pH. Some are working in acidic pH, whereas some are working in neutral pH environment. Still, others are working in basic pH environment. Therefore, all enzymes have not the same pH value. Therefore, they are sensitive to pH. They are sensitive to temperature. For example, our body temperature is around 37 Celsius or 37 degrees centigrade. So our enzymes in our body are work based in around this body temperature. Otherwise, beyond this body temperature, the functioning of these enzymes become decrease. Finally, they may be denatured. They are sensitive. I mean, they are specific in chemical reactions because one enzyme can catalyze one specific chemical reactions. So they are specific in a chemical reaction. One enzyme for one chemical reaction. That they are not consumed in a chemical reaction. For example, this is reactant A reacts with reactant B in a chemical reaction which is catalyzed by enzyme, for example, this enzyme. And at the end, product C is produced, but enzyme, the original enzyme is obtained back. Therefore, at the end of chemical reaction, they are ready for another the same catalyze the same chemical reaction. So they cannot be consumed in a chemical reaction. These are some general properties of enzymes. This is about chemical nature and classification of enzymes. All enzymes are proteins. All enzymes are proteins in nature, but not the reverse. all enzymes, all proteins are not enzymes, but all enzymes are proteins. Some enzymes require presence of cofactors in their catalytic activities, even if enzymes are proteins, but still some enzymes needs non-protein part called cofactor to be functional enzymes. Cofactors are either organic molecules such as coenzymes and inorganic molecules such as metallic ions. Cofactor can be coenzyme, and can be metallic ion. Here, the holoenzyme, which is functional in the active enzyme, is made up of apoenzyme, the protein part of this active enzyme, and the cofactor, the non-protein part of the enzyme. Holoenzyme is active in the functional enzyme. Apoenzyme is a protein part of an enzyme, as well as this cofactor is non-protein part of an enzyme. The cofactors are two types. Tightly bound cofactors are known as prosthetic groups. These are metallic ions more. They are metallic ions, and cofactors that are bounded and released easily are known as coenzymes. So cofactors can be metallic ions that are tightly bound with the protein part of enzyme, and they are known as prosthetic group. Whereas cofactors that are bound to apoenzyme or the protein part of enzyme and that can be easily released are coenzymes. These coenzymes are mostly so these coenzymes are vitamins. They are vitamins. Based on differences in chemical nature, enzymes can be simple enzyme, conjugate enzymes, metalloenzymes, and as well as isoenzymes. This can be categories of enzymes based on the nature of, based on their difference in chemical nature. Simple enzymes are enzymes that are made up of only protein, or they are polypeptide. They contain no chemical groups other than amino acids. These simple enzymes are made up of only amino acids, and they have no any other additional molecules. For instance, digestive enzymes such as pepsin and renin are simple enzymes. Conjugate enzymes are holoenzymes. Previously, we tried to discuss about holoenzymes. This type of enzymes are made up of two parts. The protein part, which is known as apoenzyme, and the non-protein part, which is known as cofactor. This cofactor can be coenzyme or it can be metallic ion. The other group of enzymes is metalloenzyme. This type of enzymes are that has metallic cofactor, which are monovalent like potassium ion, and divalent cations, for instance magnesium ion, manganese ion, as well as cupric ions. This can be used as a cofactor by conjugated with apoenzyme part of an enzyme. They make the metallic metalloenzyme. Isoenzyme are enzymes that differ in amino acid sequence but catalyze the same chemical reactions. So these are some groups or categories of enzymes. Here, enzymes is another way of classifying enzymes based on the substrate they act upon. Enzymes can be classified mostly based on their substrate they act upon, and in general there are 6 major classes of enzymes. Based on this criteria, enzymes can be classified into 6 major classes. For example, the first one is oxidoreductase. Oxidoreductase are a group of enzymes that catalyze oxidation reduction reaction. In oxidation reduction reaction, hydrogen, oxygen, and electrons are added or removed from a substance. So, for example, dehydrogenase oxidase are some examples of such enzymes. The second class is transferase. In these enzymes which are classified under this class are catalyze the transfer of special groups or functional groups such as phosphate, methyl from one substance to another. For example, transaminase, kinase are examples of this group. Hydrogenase is the third class of enzymes. So enzymes which are grouped under hydrogenase are catalyze a chemical reaction of hydrolysis. Hydrolysis means breaking down of a substance by addition of water. Therefore, enzymes that are grouped under hydrolase are catalyze hydrolysis reaction. For example, esterase and the digestive enzymes are a good example. The fourth class of enzyme is isomerase. Enzymes that are grouped under isomerase are catalyze a chemical reaction known as isomerization. Isomerization means, isomers means molecules with same chemical formula but different in structure. For instance, glucose and fructose have the same chemical formula, but they are structurally different. Therefore, for instance, glucose can be converted into fructose. So this reaction is known as isomerization reaction. So this is catalyzed by isomerase enzymes. Phosphohexoisomerase, fumarase are some examples. Lyase is another class of enzymes. This lyase catalyzes a chemical reaction, non-hydrolytic removal of a group or addition of a group to a substance. Groups may be removed or added to a substance without hydrolysis. So such chemical reaction can be catalyzed by lyase. For example, decarboxylase, an enzyme used to remove carbon dioxide from a substance. Aldolase is another example. Ligase is the 6th class of enzyme. Ligase or synthesis. Such enzymes can catalyze the joining of 2 molecules by the formation of a new bond. Whenever smaller molecules come together, they make new larger molecules with the formation of new bonds. Therefore, such chemical reactions can be catalyzed by ligase or synthesis enzymes. For example, citric acid synthase are example. In general, oxidoreductase, transferase, hydrolase, isomerase, lyase, and the ligase are the 6th class of enzymes. Another point is mechanism of enzyme actions. In principle, all mechanisms of enzyme actions are reduction of energy barrier which separate reactant from their product. In a chemical reaction, there is a reactant which can be converted into the product. However, there is energy separation or energy barrier that separates these 2 parties. So the activation energy is needed by the reactant molecule to overcome this barrier and form the product. The reactant must get activation energy to get or to be overcome or to break the barrier and form the product. So what is the activation energy? Activation energy is a type of energy which is needed to start a chemical reaction. So these reactant molecules must get this energy, the activation energy, to get started the chemical reaction. Then the product must be formed. For example, the matchbox and the matchsticks are found together, but there is no formation of fire. However, whenever we apply some friction force, immediately fire will be formed. Therefore, after applying some activation energy, the reactants cannot be react for the product. So the main function of enzymes is to produce the amount of activation energy that is needed to start the chemical reaction. So once the chemical reaction is started, then the reactant will be going on and the product must be produced. So enzymes are required to reduce the amount of energy, the amount of activation energy that the reactant needs to get reacted and the product to be formed. The substrate of enzymes are reactants of a chemical reaction that are catalyzed by enzymes. In a chemical reaction, there is reactant that can be converted into product. So this chemical reaction is catalyzed by an enzyme. So these reactants of a chemical reactions are substrates of an enzyme. For example, here sucrose is a reactant in a chemical reaction that produce glucose and fructose. That is the conversion of or the breakdown of glucose into sucrose into glucose and fructose is catalyzed by an enzyme. The active site of an enzyme and the shape of the sucrose are complementary. So in this way, reactants of a chemical reaction or substrate of an enzyme can be converted into the product. Enzymes are specific to their substrate, meaning one enzyme for one substrate. This is determined by the active site of an enzyme. Active site of an enzyme is an area on the enzyme where substrate binds. Even if an enzyme has different parts, but an enzyme, a substrate is bind to the active site of an enzyme. The combination formed between the enzyme and its substrate is known as enzyme-substrate complex. Enzyme combined with substrate, so this enzyme-substrate complex is formed. Substrates are attached to the active site by electrostatic and hydrophobic force, which are non-covalent bonds, or they are physical attraction. They are not chemical attraction. Therefore, they can be easily dissociated at the end of the reaction. Therefore, in this way, this enzyme substrate complex is dissociated into enzyme plus product. Enzyme plus product. Therefore, the force that associate this enzyme and the substrate are electrostatic force as well as hydrophobic forces, which are physical attractions that can be easily broken down. There are two theories about how enzymes and the substrate interacts or bind. There are two theories. The first theory is a lock and key model theory. In this theory, an enzyme is like a key fitting to a lock. The shapes are fixed. Neither the lock nor the key shape change their shape. There is no change in shape of the lock and the key. Already their shapes are fixed. Therefore, the shape of the active site of an enzyme and the shape of the substrate are unchanged. It is fixed. This theory proposes this idea. But in induced fit model theory, the substrate doesn't simply bind with an enzyme at its active site. Rather, enzyme's active site should be changed in its shape to be complementary and bind with the substrate and make the reaction possible. In this theory, to some extent, the active site of an enzyme to be changed its shape to bind with the substrate. To make a complementary and bind with the substrate, it must change its shape to some extent. So this is known as induced fit model theory. These are the two main theories of mechanisms of interactions of enzymes with its substrate. Another point is factors affecting enzyme activities or enzymatic activities. There are factors that affect enzyme action, for example, temperature, pH, substrate concentration, enzyme concentration, as well as presence of inhibitors are some factors that affect enzyme actions. Temperature. Increasing temperature increases kinetic energy of molecules. Whenever the temperature increases, the movement of molecules increases. This means that there is more random collision between molecules. Whenever molecules are moving here and there faster, there is a chance to be collide to each other. However, increasing temperature at a certain limit also increases vibration energy of molecules, in this case enzymes. So this puts damage on the bonds that hold them together. If the bond is broken down, especially the active site of, the shape of the active site of an enzyme will be changed. So active site is less complementary to its substrate. So there is no chance to be join the active site and the substrate. So it affects, increasing temperature at certain limits affects the binding of the active site with the substrate. Increasing temperature at certain limits not only affects the chance of active site of an enzyme to bind with substrate, but also eventually the enzyme itself will become denatured because enzymes are proteins, so they have tertiary structure. So when the temperature increases, they lose their tertiary structure, meaning they will be denatured. And eventually, at the end, they cannot be functional molecules. The temperature at which maximum rate of reaction occur is known as enzyme's optimum temperature. Each enzyme has a temperature range or optimum temperature in which maximum rate of reaction is achieved. For example, we human beings body temperature is around 37 degrees centigrade. At this body temperature, there is maximum rate of reaction is recurrent. But if the temperature is down or decrease, similarly rate of reaction decreases. When the temperature decreases, rate of reaction decreases. Even as at some point the reaction becomes zero. If the temperature is increased, from 37 to above, the rate of reaction still decreases, and at some point it becomes 0. At this point, there is the nature of the enzyme. So the enzymes are not functional at their denatured condition. Similarly, enzymes in cold water shrimps, these organisms, their maximum rate of reaction is recorded at a temperature of 40 degrees centigrade. But when their body temperature decreases, the rate of reaction decreases too. When the body temperature of this organism increases, rate of reaction also decreases. Here, enzymes in bacteria living in hot spring, their body temperature, their body temperature is around 95 Celsius or degree centigrade, at which maximum rate of reaction is recorded. But when their body temperature decreases, their rate of reaction decreases too. When their body temperature increases, rate of reaction decreases. Therefore, temperature affects rate of reaction by affecting the chance of the binding of active sites of an enzyme with a substrate, as well as gradually it may destroy the tertiary structure of an enzyme and the enzyme become denatured. pH value, that means acidity and the basicity of the solution, also affect the enzyme action. pH is a measure of hydrogen ion concentration in a solution, and also is an indicator for hydroxyl ion concentration. Low pH, that means the pH value is from 14 to 1. Here at the middle, 7 is neutral. Above pH value above 7 is basic, whereas pH value below 7 is acidic. Therefore, in low pH, there is high hydrogen ion concentration and low OH ion concentration. Enzymes have different optimum pH. Optimum pH value is the pH value of an enzyme at which active site of an enzyme is most complementary to the shape of their substrate. That means when the pH is changed from its optimum, immediately the active site of an enzyme, the shape of the active site of an enzyme will be changed. Therefore, at optimum pH, there is high chance of active site of an enzyme and the substrate can be joined together. At optimum pH, the rate of reaction is maximum. At optimum pH, there is a great chance of the binding of active site of an enzyme and the substrate, so there is maximum rate of reaction is occurred. For example, in our body, there are different pH values are found. If we take the digestive system, the pH value in the mouth is different from the pH value in the stomach, and it is different from the pH value of the small intestine. Therefore, the optimum pH of enzymes in each of these sites is different. For example, pepsin, an enzyme which is active in stomach, is active. Its optimum pH is around 2. Therefore, there is high activation energy, high rate of reaction is occurred at the pH value of the pepsin, which is its optimum pH value. Whereas trypsin is another enzyme. It is optimum pH value is around 8. So maximum rate of reaction is found at this optimum pH value. Therefore, in the same organism, there are different pH values or optimum pH values. Therefore, there are different enzymes are found at such optimum pH value to catalyze faster or maximum rate of chemical reactions. Any change in pH above or below optimum will quickly decrease in rate of reaction because shape of the active site of an enzyme is not or less complementary to the shape of their substrate. Small change in pH doesn't permanently change the shape of the active site of an enzyme, since the bonds are the bonds can be reformed. However, extreme changes in pH can cause enzymes to denature and permanently lose their function. Enzymes in different locations of our body have different optimum pH value. Substrate concentration is a third factor that affects enzymatic activities. Increasing substrate concentration increases rate of reaction with constant amount of enzyme concentration. With the same or constant enzyme concentration, whenever substrate concentration increases, rate of reaction increases. This is because substrate molecules will be collide with enzyme molecules, so more products will be formed. However, after certain concentration, any increasing substrate concentration will have no effect on rate of reaction because all enzymes will become saturated and they will be working at their maximum rate. Here, for example, substrate concentration increases. Similarly, rate of reaction or velocity increases. Substrate concentration increases, rate of reaction increases. But at certain limits, a certain point where all the enzymes are occupied, then rate of reaction becomes constant. Rate of reaction becomes constant because all the enzymes are saturated and they are working at their maximum rate. Therefore, at Vmax, after Vmax is attained, whenever substrate concentration is increased, rate of reaction becomes constant. Enzyme concentration is another factor that affects enzyme catalytic action. Rate of reaction will increase if enzyme concentration with substrate concentration increase, because both enzyme concentration and substrate concentration increase, so rate of reaction increases because there is a chance of collision of enzyme molecules and the substrate molecules, so there is increase on a chemical reaction. The fifth factor is enzyme inhibitors. These enzyme inhibitors are molecules or chemicals that inhibit or discourage the enzyme activities. Enzyme activity can be inhibited or discouraged in various ways. These enzyme inhibitors are chemicals and that reduce rate of enzyme catalytic action. They are usually specific. Specific means each inhibitor is for a specific enzyme. One enzyme for one inhibitor, and that they work in low concentration. To inhibit a chemical reaction that is catalyzed by enzymes, these inhibitors can be found in lower concentration. This is the two nature or characteristics of inhibitors. They are specific, that means one inhibitor for one enzyme, and they are needed in low concentration to be obstacle to inhibit enzyme action. Inhibitors could be reversible, meaning they can reverse, the enzyme that is inhibited by such enzymes can be reversed to its normal function, and some may be irreversible. Enzymes that is inhibited by irreversible inhibitors cannot be returned to its normal state or normal functioning ability. These reversible inhibitors are competitive inhibitors. The substrate and the inhibitor are competing for the same active site of an enzyme. As the name shows, they are competitive because they compete for the same active sites with the substrate, and still there are non-competitive. Non-competitive inhibitors are not competing with substrate with the same active sites of an enzyme. Reversible inhibition, which is competitive as well as non-competitive inhibition. Competitive inhibition occurs when inhibitors are similar with the substrate molecules and compete for the same active site of an enzyme. The two molecules, substrate as well as the inhibitor, are looking for the same active site of an enzyme. So this inhibitor will compete with the substrate for the same active site. So it prevents substrate from binding to the active site. For example, penicillin, which is antibiotic given for bacterial infection, is a competitive inhibitor that blocks the active site of an enzyme that many bacteria use to construct their cell. As you know, bacteria have cell, so this penicillin is used as a competitive inhibitor for many enzymes that are found in bacteria that are used to construct cell of the bacteria. So if bacteria lose or have no cell, it can be easily destroyed, because if water molecules are enter into bacteria, so easily they can be disintegrated. Therefore, in this way, bacterial infection can be prevented by penicillin. This competitive inhibition can be reduced by increasing concentration of substrate, because the chance of a substrate to bind the active site increases whenever the concentration increases. Non-competitive inhibition occurs when inhibitors bind to the enzyme at a location other than the active site called allosteric site. This allosteric site is a site on enzyme other than active site, and the inhibition is known as allosteric inhibition. These inhibitors in non-competitive inhibition are looking for the active site of an enzyme. Rather, they look for the allosteric site. Whenever the inhibitors are bind to the allosteric site of an enzyme, immediately the active site of an enzyme will lose its shape. So the substrate can't bind to the such active sites. This inhibition, meaning non-competitive inhibition, cannot be influenced or overcome by increasing the concentration of the substrate, because a small amount of non-competitive inhibitors can easily affect the enzyme catalytic actions, because they are looking for the allosteric site, not the active site of an enzyme. But the substrate is binding to the active site. But once the allosteric, the non-competitive inhibitors are binding to the allosteric site, immediately the shape of the active site of an enzyme is changing, so the substrate can't have a chance to bind these active sites of an enzyme. End products of a metabolic pathway serve as allosteric inhibitors. For example, this is a metabolic pathway where reactant A is reacted and produce product B. The first chemical reaction is catalyzed by enzyme 1, and this product before first reaction can be reactant for the second reaction. So this B is converted into product C. This chemical reaction is catalyzed by enzyme 2, and the product of reaction 2, C, is or can be reactant for reaction 3, and then the reaction 3, this C is converted into product D, which is the final product, and then the third reaction is catalyzed by enzyme 3. In the product inhibitor, in the product of a metabolic pathway that serves as allosteric inhibitors, this product D will bind to enzyme 1, the first enzyme that catalyzed the first chemical reaction allosterically. Then after, the whole pathway will be blocked. So there is no change from A to B, then B to A, C, C to D. Whenever product D is required, immediately this enzyme 1 will be free, and the pathway will continue. In this manner, the metabolic pathway can be controlled by allosteric inhibitors. The inhibition of an enzyme by the product itself. The product itself is the form of negative feedback, because the product of a pathway can be used as inhibitor. It inhibits, or the product inhibits, the first enzyme that catalyzes the first chemical reaction. So this is negative feedback. Another factor is activators. Inhibitors inhibit enzymes activity, whereas activators activate or enhance enzyme actions. An inhibitor, an activator molecule can be bound to allosteric site and enhance a reaction at active site by changing the shape of its substrate. Common activators include hormones and the products of their enzymatic reactions. So activators can enhance enzyme catalytic activities. The other factor is reversible inhibition. In reversible inhibition, inhibitors are bound to the enzyme, and the enzyme will be malfunction forever, even if the molecules, these inhibitor molecules, are removed. Irreversible inhibitors are generally specific for one class of enzyme and do not inactivate all enzymes. They do not function by destroying the enzyme structure, but specifically they altering the active site of their target enzyme irreversibly. So irreversible inhibitors often contain reactive functional groups because the active site of the enzyme is made up of different molecules. Therefore, this functional group of the irreversible inhibitors can change the molecules in active site of the enzyme irreversibly. Therefore, once this the active site of an enzyme is irreversibly changed, such enzymes are malfunctioned forever, even if the inhibitors are removed. Thank you for watching. I will come back soon with the second part of this chapter. Please don't forget to like, share, and subscribe this channel. Thank you once again.